Hi Professor and classmates, my name is Minh. I'm from Plant Immunology Lab. So in this presentation, I would like to present to you plant immunity research in our lab and also what I have learned so far. Let's start the first part, introduction. I would like to talk about the importance of plant immunity. During the growth, the plants are exposed under many kinds of stress. Abiotic stress contains heat, cold, drought, flood, and so on. In case of biotic stress, there are bacteria, virus, and pests. They can affect to the plant and cause the disease to the plant. In worldwide, 14.1% of crop lost due to the plant disease. So you can imagine if the plant disease increase, the biomass production will be decreased. And many potential biotic stress can affect to the plants, but only limited number of pathogens can successfully invade into the plant. So plants need to develop the defense system to protect themselves. So what is the plant defense system? The plant defense system contains preformed defense and induced defense. In preformed defense, plants have a cell wall and some metabolites. They are the first barrier of the defense. But if this first barrier is not strong enough against the pathogen, plant will activate the induced defense. Induced defense contains PTI, which is the pathogen-associated molecular pattern triggered immunity, and ETI, which is effector-triggered immunity. So I would like to explain about PTI and ETI. In PTI pathway, pathogens contain some special structure called PAM, and PAM can be recognized by plant receptor PRR. When PRR recognizes PAM, PRR can signal downstream to activate some mechanism such as ROS production, antimicrobial compounds, and activation of defense genes in nucleus. However, pathogens contains another weapons to deal with this problem. They deliver effectors protein through type 3 secrete system. The function of effector is to suppress the PTI signaling pathway. But plants also have the defense system called resistant protein. Resistant protein is the RLRs. These RLRs can recognize the presence of effector directly or indirectly. The outcome is that the plants can cause the hypersensitive response or the cell death to protect themselves against the pathogen invasion. Then why plants need the cell death? This is because if the bacteria invade into the plant can be diseased or resist against the pathogen. If the plant resists against pathogen, the leaf seems very healthy, no disease symptoms. However, if the number of bacteria is too high, the plants recognize this problem and cause the localized cell death. This localized cell death is happen only in certain area to protect other area from the invasion of pathogens. Next, I would like to talk about my research purpose. So my study is about an effector called AVR-RPS4. When AVR-RPS4 is secreted into the plant cell, it can be cleaved into two parts, N-terminus and C-terminus. Previously, people demonstrated that only C-terminus can target to the working domain of RS1, lead to the activation of pair R protein, RPS4 and RS1, then resistance happen. Another research group proved that both N-terminus and C-terminus can somehow target to EDS1 and lead to the resistance. However, the mechanism how AVR-RPS4 is recognized by plant is still unknown. So the purpose of my study is to answer the questions how AVR-RPS4 recognized by plants and what are the targets for AVRPS4 N terminus in the plant. During the studying in the lab, I have learned so much things, such as the communication with professor and lab members, the cooperation, working as a team, and management. And also, besides those things, this presentation I mainly focus on methods, what I have learned so far. This slide is about the pathogenesis assay. To do this experiment, first I need to generate the transgenic plants. So I need to clone my gene of interest into the vector and transfer the vector to the plants and select the homolines. After I got the homolines, I can do the infiltration method, in which I deliver the bacteria through the syringe into the leaf tissue, and later I can count the number of bacteria in the leaf tissues. If you can see on the right side, this is the graph showing the number of bacteria in wild type and transgenic plants. The number of bacteria in transgenic plants seems reduced compared to the wild type. And if my gene of interest is expressed in these transgenic plants, plants recognize this problem and cause a resistance. So, by using the pathogenesis assays, I can say that my effector can somehow cause a resistance to the plant. And one important thing is that by using this experiment, I can know which plant is resistant, which plant is susceptible to the pathogens. The next method I would like to introduce is hypersensitive response, or HRSA. The purpose of this experiment is to determine whether the plants can recognize the effector or not. If the plants recognize the effectors, cell death happens. If not, no cell death. So to perform this experiment, first we need to prepare the plants and also the agrobacterium containing our gene of interest. And next we can make the bacterial solution and inject that solution in the leaf tissue. And after 1, 2, or 3 days, we can analyze the HR symptom by eyes, or we can use the UV box, or we can quantify the HR level by iron leakage assay. In here, I only show the picture of the electric lips under the UV box, and I perform the HR assay in here. The green color represents for the HR symptom. In that spot, the plants recognize the presence of effector and trigger the cell death. The next method I apply to my study is the yeast to hybrid screening to find effector targets. You can imagine this is like fishing. If you use the bait, you can catch the prey. So in my case, I use my effector as a bait, and I can find some interactor protein from the whole library. So this experiment based on the yeast system. We can fuse protein X with the binding domain and protein Y with the activation domain. If X and Y do not interact, the activation domain cannot recruit the RNA polymerase to activate the reporter gene, so that the yeast cannot survive on the selection media. However, if X and Y interact, activation domain can recruit the RNA polymerase and activate the reporter gene. The outcome is that yeast can survive on the selection media. If I find any interaction in the yeast, I can confirm that interaction in planta. BIFC is one of the methods to determine that interaction. The principle of BIFC is based on the yellow fluorescent protein emission signal. In this experiment, we can fuse protein A with N terminus of YFP and protein B fused with C terminus of YFP. And we can express both constructs in plants. If A and B interact, N terminus of YFP is close enough to C terminus of YFP and emits the signal. And we can see the signal under the confocal microscope. So to perform the BIC experiment, first we need to prepare the tobacco plants and also the agrobacteria containing our gene of interest fused with N or C terminus of YP. And next step, we need to mix both constructs to make the agrobacterial solution. And later we can inject that solution in the tobacco leaf to express that protein. And later, after 2 days, we can analyze the biopsy signal by using the confocal microscope. In here, I attack the example of no interaction and the interaction. If we cannot see any signal from the microscope, we can think that two proteins do not interact. And if we can see any signal under the microscope, we can think that they interact to each other. The interaction can be in nucleus, cytoplasm, or the membrane. And in this case, the yellow spot indicates the interaction happened in the nucleus. Another method to determine the interaction in plants is the co-IP. The principle of co-IP is that if X and Y interact and we can pull down X, which means that we can pull down Y also, and we can see the presence of Y in the Western blot. If we cannot see the presence of Y, which means that X and Y do not interact. First, we need to prepare the tobacco and also the agrobacteria containing our gene of interest. And step 2, we need to mix the bacterial solution containing two constructs, and we inject that solution in the tobacco to do the transient expression. Step 4, we need to extract the protein, and for example in here there are two protein fused with two different tags, flag tag and the mid tag. And next step, the IP will perform. For example, if we use the bead, agar bead containing the anti-MYC antibody, we can pull down only protein fused with the MYC. So if the protein fused with flag interact with the MYC fused protein, that protein can also pull down by the bead. And after watching step, we do the co-IP Western blot, and if we can see the flag signal in the anti-flag blot, which means that there is the interaction. If we cannot see any signal, which means that no interaction. Protein interaction confirmation is not enough for the study, so I need to make the knockout overexpression and complementation lines of gene of interest and check the pathogenesis assay in that plant to know the biological function of that protein. This is the final slide of my presentation. Thank you.